you, Alan, for the very kind introduction and for the opportunity to speak here at the Platelet Society. It really is an honour to be invited to give a public lecture on a topic that has affected every single one of us at all parts of the globe. We have all become familiar with images like this of the coronavirus. And one of the things that I feel has really become evident throughout this whole pandemic is the fact that science is now embedded in our general community. And so us as scientists should feel proud with the fact that now our voices are being heard much more readily. So I'm trying to look on the positives because obviously there has been quite a number of negatives. The SARS-CoV virus first afflicted us in November 2019. It originated in Wuhan, China, and from then it became a worldwide pandemic that we are now all too familiar with. You can see here from the WHO data that we have different countries have been affected by this virus to varying degrees, but one must remember as well that often the incidence of SARS-CoV infection, of which the disease is called COVID-19, are not recorded in the same manner by different countries. And so this can have an impact on the absolute numbers in terms of what we're looking at. But nevertheless, what you can see is that westernized populations have been very badly affected by this virus. So these are images that we are now familiar with seeing on a daily basis. We know how many people we're allowed to meet up with, although that can get confusing considering that we have different rules in different parts of the UK. We know that we're meant to stand 2 meters apart and also washing our hands, wearing masks while out and about, but also the more sad images in terms of seeing our doctors and nurses having such a struggle in ICU units to tend to patients with severe COVID-19 infection. Obviously now we have mass testing available to us, and again this is becoming part of our daily lives. Those of you who homeschool will also be familiar with this sort of image. This is one of the ways that the pandemic and the ensuing lockdown affected all of us, but one of many, from having to work alongside children at home or other caring responsibilities, to not being able to see friends and family, and not having those reunions and those things that as a human a race really set us apart in terms of enjoyment and socializing. Another thing I was just going to mention is the hairdresser. Obviously we don't have that either. So back to COVID-19 as a disease. So COVID-19, as you'll all be familiar from the information that comes out to us from the public broadcasts, is associated with fever and fatigue and a dry cough. And these are the symptoms. Loss of taste as well as was added in as ones that we needed to watch out for. But what you can see from this timeline here is that the initial symptoms can develop. Now, some people are asymptomatic. They don't even have these very minor symptoms of COVID-19, and that is one of the complexities of COVID-19 as a disease. However, as the days progress on, if an individual is going to be predisposed to developing severe COVID-19, they develop a shortness of breath, a dyspnea, which can, under some circumstances, lead to hospital admission. And you'll see here that the average timeline for that is around 8 days. And then what is again surprising is that even when an individual is admitted to hospital, they don't necessarily develop a severe disease. That can be 4 or 5 days later, up to 7, 10 days later, once they have been admitted to hospital. And that is when we see ICU unfortunate events in terms of numbers rising, and that has been a critical component in terms of the number of ICU beds available. So what is COVID-19 pneumonia? As I said, most patients with COVID-19 do not develop a pneumonia. However, those that do, it can be characterized quite clearly by various types of appearances in the lung. These have been called ground glass opacity, and then they're followed by linear opacities and consolidation. You can see this chest X-ray of this 50-year-old man on the right, and this is actually an unusual circumstance in that the man had a chest X-ray 12 months prior to developing COVID. So this individual at this point does not have COVID, and you can see the changes. This is called day 0, but this is day 0 on admission to hospital, and as I just noted on the previous slide, that can be some days after development of the disease. And then at day 10, when the individual is in ICU, again, you can see appearance of these ground glass opacities, which are prevalent generally in the peripheral and lower regions of the lung and mostly appear bilateral. There has been some incidences of them being unilateral in the lung. These are also evident on CT scans. But we now know that COVID-19 is not just a respiratory disease. It can affect many different aspects of the human body. And in fact, we're only at in our infancy in uncovering the number of different ways that the SARS-CoV virus can impact on the organs of the body. It has to be said, though, that there is a huge body of literature that has developed over the past year, and one that is really quite remarkable in terms of the science world being able to attack essentially what is going on and try to understand the complexity of this viral infection. I have to add a disclaimer at this point that I will be focusing on the coagulopathy associated with COVID-19, but even that is a wide body of literature and one that cannot be covered in a short presentation as this. So I've had to cherry pick a few of the studies which I find of interest, but there is many more studies out there which have helped develop our understanding of SARS-CoV infections. So how does the virus get into our body? That is now well understood to be via the angiotensin converting enzyme receptor. And this ACE2 receptor is prevalent on epithelial tissue. What happens is that the SARS-CoV virus, which is shown here with its characteristic spike proteins, can attach to the ACE2 receptor. And in combination with transmembrane serine protease, it can then endocytose into the cell, and as a result of that can replicate within the cell and thereby reproduce, therefore increasing the number of virus that are present in our body, what is called viral load. As I showed you on the previous slide, a number of organs are affected by SARS-CoV-2 infection. And that is perhaps not surprising when we look at the expression of the ACE2 receptor throughout our body. You can see here on this sliding scale, with purple being the highest expression of the ACE2 receptor, that there are a number of tissues, obviously including the upper airways and the lungs, which we would expect, that have very high levels of the ACE2 receptor. And I should say that the ACE2 receptor is involved in regulating blood pressure, and as a result of that is prevalent in tissues such as the lungs and the cardiovascular system. What are the hallmarks of COVID-19? Again, I'm really only going to touch on this as a various aspect in terms of outlining what is potentially, is the underlying factors as to how this infection can really take a grip of the body very, very quickly in some instances. And what happens is that when the virus enters the cell via the ACE2 receptor, it's then able to increase its number, and these are subsequently released by the cells. But this damage to the endothelium by the virus then results in a huge inflammatory response. And what we see is that we have dramatic augmentation of a number of different pro-inflammatory cytokines, such as IL-1, IL-6, and TNF-alpha. There's also an increase in hypoxia within the lung, which results in a decrease in gas exchange. So there's less oxygen, hence the symptoms that we classically associate with a severe COVID-19 infection. There's buildup of fluid, and the onslaught in terms of the cytokine storm that occurs causes dramatic inflammation in the surrounding tissue. But as I said, we know that the COVID-19 disease does not only affect the lungs, and we also know that there is risk factors that are associated with severe COVID-19 infection. What's not surprising, but nevertheless it is something to really remember, is that the number of the risk factors for COVID-19 infection are actually risk factors for cardiovascular disease as well. So we know that age is a risk factor for COVID-19, and this was very evident in the early stages where we saw rampant SARS-CoV-2 infections within care homes. But we also know that now that there are other factors which can predispose individuals at higher risk of severe COVID-19 disease. And these are classically now described as hypertension, diabetes, obesity, prior heart disease, thrombotic disorders, and ethnicity. So, as I mentioned, the impact of COVID-19 is not only related to the lungs, and it's not only related to the inflammatory side of the pathways. These are quite gruesome images on the left-hand side here, and I apologize for those that I should have given a prior warning just to the slide that was coming up. But we can see that we're on the right, we're looking at various tissues from various parts of the body. We have tissue from the pulmonary artery with surrounding lung tissue, tissue from the kidney, and the cardiac tissue and lung. And in all of these organs, what we see is evidence of thrombosis, whether that be micro or macro thrombosis, which is basically occurring systemically throughout the body. Now, the incidence of thrombosis is particularly high, and I'll come on to that in a minute, despite the fact that often these individuals are on prophylactic or preventative thrombolytic anticoagulant therapy. And you can see that one of the most common sites, perhaps not surprisingly so, for development of these thrombi is in the lungs, and these are described as pulmonary emboli. What's interesting is that, and has just come to light in the last few months, is that perhaps these pulmonary emboli are dissimilar to the pulmonary emboli that we would normally see, and that they are often formed within the lung themselves rather than moving from a distal part of the body. So as I said, the incidence of venous thromboembolism, which is largely what we'll focus on today, so that is thrombi that form in the venous circulation, is approximately 30% in COVID-19 patients. And the mortality rate of those COVID-19 patients that develop the VTE is higher. You can see down here some statistics that around 19% will develop deep vein thrombosis. So that's when the thrombi form within a vein, usually in the leg, but can also be the arm. Deep vein thrombosis is classically associated with pulmonary embolism, and collectively, these two types of thrombosis make up the syndrome called venous thromboembolism, or VTE. There is also incidences of arterial thrombosis. These are more rare, but nevertheless do exist. So I now want to talk about what tips the balance in COVID-19. Why are we predisposed to developing thrombosis with this infection? So in this beautiful diagram that was published in the Lancet by O'Sullivan, we can see what is on the right-hand side a normal alveolar in the lung. And you can see that we have movement, obviously, of all the factors that are important in our blood in clotting. And clotting is inherently important, as we know. Without adequate hemostasis, then one would not survive. But nevertheless, when it becomes aberrant, is when we have thrombotic complications. What you'll see here is that there is an endothelial lining within this alveolar, and this is continuous throughout the vasculature, and it is extremely important in terms of maintaining blood fluidity, because it hosts a number of different factors which dampen down any procoagulant response. It contains nitric oxide, which will basically keep platelets at in a resting, more quiescent state, and it also contains a number of anticoagulant proteins which line the surface, such as thrombomodulin thrombomodulin. If this surface becomes damaged in any way, then we see what's called rupture in terms of the barrier of the endothelial surface, and as a result of that, these factors that are circulating in the blood can come in contact with underlying prothrombotic factors. This can result in development of a thrombus, as shown here on the left-hand side. So what happens is this cytokine storm that we talked about basically causes endothelial disruption, and this disruption to the endothelium goes hand in hand with development of thrombotic complications. We're now going to go through some of these thrombotic complications that occur. And one of the things that became evident immediately is that the individuals with COVID-19 have a hypercoagulability. So basically, that's when their clotting factors are activated. And very quickly, D-dimer was noted as being a factor that was increased in COVID-19 and has since been used. It was originally denoted by Tang, but there's been multiple other publications that have shown that it is a measure. But we also know that as well as D-dimer, there are other factors such as factor 8 and von Willebrand factor and fibrinogen that are elevated. And that goes hand in hand as well with an increase in microvesicle and monocyte tissue factor. As well as the hypercoagulability, we see depression of fibrinolysis. So that is the opposing system which counteracts the clotting system. And again, we can see that there is changes in the fibrinolytic activity. These include increases in an inhibitor called PI1. This is a recent publication by Zhu et al., which has detailed this very nicely. And as a result of that, we also see correlations with the D-dimer and depression of overall fibrinolytic function. This elevated PI1 expression is perhaps not surprising, given the fact that PI1 is elevated in a number of the different risk factors for cardiovascular disease and also inflammatory diseases such as obesity, type 2, and infection. Neutrophil extracellular traps have been implicated in the development of the prothrombotic complications. These are nets that are extruded from neutrophils, a type of leukocyte. This was first exposed by Brinkman et al. In 2004, and we now know that they play a large role not only in controlling infection, but can also promote clotting by binding various coagulation factors and also platelets themselves. And these very nice images here with normal plasma and with COVID-19 plasma show that in COVID-19 plasma, there is much higher incidence of development of nets than in normal plasma. And there has been some beautiful publications. Again, there's another one by Zhu et al. In 2020 that shows that there's a direct correlation between factors that or markers for neutrophil activation and the development of coagulopathy. As I said, nets are also enriched with coagulation proteins, and there has been, again, links in the literature between the coagulation and the complement proteins. I can't go past this without mentioning platelets, and I'm not going to talk very long about it because this whole meeting is focused on platelets, but nevertheless, platelets are also very important in this inflammatory response. And one of the ways that platelets can act is by interacting with neutrophils, and this is well established in the literature now. And indeed, there are now publications that show that platelet activation is prevalent in severe COVID-19 disease. And what we see, this is platelet spreading down here in the absence of COVID and in the presence of COVID. And we see enhanced spreading and also enhanced platelet aggregation. But the platelets will also interact with neutrophils and monocytes to further augment this prothrombotic changes that are occurring in COVID-19. So this concept is called immunothrombosis. It's basically an interaction between the hemostatic system and the innate immunity system. And as I've shown you in the previous slides, there's many cellular elements that are involved in this, including neutrophils and monocytes and, of course, platelets. And really, we're still in our infancy in understanding all of the different interactions between immunity and the hemostatic system. What are our treatment options? These are varied in terms of talking about specifically the prothrombotic complications, and there are studies going on worldwide that are looking at various types of treatment. These three studies here, Active-4, ATTACK, and REMAP-CAP. Actually, they have many studies. The Active and the REMAP have many studies going on, but the 4 specifically relates to antithrombotic studies, and one of the aspects or the arms of the REMAP is also an antithrombotic study. But what is nice is that these studies have come together. They have aligned their protocols and their outcomes such that we can hopefully get a better understanding of how to treat patients with prothrombotic complications in COVID-19. And you can see here that one of the markers that we talked about initially is D-dimer level. And the interim results of these studies have just been published, and what they show is that therapeutic anticoagulation is beneficial in dampening down the prothrombotic response compared to the prophylactic treatment that these patients were receiving. So these studies have focused on heparin. Heparin can potentially have a pleiotropic action in terms of treating COVID-19. This includes the anticoagulant action that we talked about, but it is also postulated that it could be antiviral in that it would inhibit viral entry into the endothelial cells and will also dampen the inflammatory response. So there may be many different points where heparin can intercept the SARS-CoV-2 infection. But of course, we could also target the inflammatory cascade, and there are many studies that are also focusing on this. The recovery study stemming from Oxford has focused on various types of inflammatory. They were the first to show the usefulness of desmopressin early on during the first wave of COVID-19, and are now looking at tocilizumab as a treatment. The initial results for this are out as well, and there is a reduction of 4% in death with treatment with this IL-6 inhibitor. So we're at a crossroads. Where do we target? Do we target the hemostatic system? Do we target the immune system? And I don't have time to go through all of the various ways that we could do this, but you will see other anticoagulants, antiplatelet drugs in there, as well as other targets in terms of the inflammatory cascade. There's other novel points of interception as well, and that includes perhaps targeting the fibrinolytic system with TPA and also targeting the contact pathway, which we haven't had time to talk about today, but can be important in that mitosis that we saw with the neutrophils extruding their nets. very final slide, apart from my acknowledgements, I felt I could not say all of this without acknowledging the vaccine-associated thrombosis. And the MHRA did reveal last week that they are confident that there is no link with the AstraZeneca vaccine and VTE complications. And this was followed by endorsement by the US. There are some reports on an extremely rare immune thrombocytopenia thrombosis, which is related to platelet activation, but this remains to be consolidated. So with that, I would just like to thank my team in the lab that are also working on the COVID side of things, and that's Dr. Claire White, Dr. Gail Morrow, Megan Simpson, and Carol Wallace. Undergraduates that have focused on COVID-19, Martin McGeddy and Grace Otterbein, which I have used some of their images in this presentation. And also our funders, NHS Grampian, of course, the British Heart Foundation, a local charity, Friends of Anchor, and Medical Research Scotland. Thank you all for listening.
Brilliant. Thank you so much for a fantastic talk, Nicola. We're getting a few questions if you're okay for a few.
Yes.
So one question that we've had is, could you comment on those apparently healthy individuals without prior cardiovascular disease? Why are some people severely affected in this apparently healthy population rather than not everyone?
That's something that requires more work, and I don't think this is really complex. We do not understand why some people can be asymptomatic and other people who appear very healthy can be very fit. They don't necessarily fall into any of the high risk groups in terms of COVID, but nevertheless will go on to develop disease, and we really do not know enough about it, despite the plethora of studies that have come out. I think this is going to be one of the ones that will take a long time. We're drilling down in terms of molecular mechanisms, which is great, but we need those long population studies to understand that.
Brilliant. We have a question here from John Gibbons. He says, fantastic talk, Nikki. Can I ask if you could comment on the timing of severe COVID symptoms? That this is around 7 to 10 days, which coincides with the appearance of anti-spike antibodies. Is this because it coincides with the appearance of the anti-spike antibodies?
Yeah, that's a great question, actually, and thanks, John. And I haven't seen any evidence on the actual antibodies that we're producing, but it could be out there, and so it's something that we should be looking at, and how that implicates changes the way our vascular response is to the virus as well. So, fantastic question.
I mean, you touched on earlier the idea that there are ways that we can break down blood clots, and obviously once they're formed. Is there any evidence that those blood clots are different to the ones we see in, say, normal heart attacks and strokes, and maybe more or less resistant to sort of those clot-busting drugs that we see?
Yeah, that's a great question. And I think one of the main things that has come out just fairly recently, and I touched upon that in the talk, and apologies, I was running over, so I did have to skip on some of the detail and some of the slides. But what we're seeing is that that the thrombi are forming very much within the lung and are perhaps not moving. And so that then gives them a very different composition as well. And I suspect that denuding of all the anticoagulant proteins that are on the surface of the endothelium will also change the platelet composition and the fibrin structure of the clot. So this will have an impact on the ability of fibrinolytics. There is some trials, again, I didn't go into these. There's a trial coming out of Harvard in terms of TPA being used as a treatment for COVID. And there's also a trial that I was also involved in coming out to the Royal Fleet as well, in terms of nebulized TPA to be able to attack the clots that form directly in the lung. But what we know is that not all the clots form in the lung, but there is a propensity for them forming there. So this will be something that we need to look at and probably require more targeted approaches.
I think we have time for one last quick one. Oh no, we've probably just run out of time. So I'm going to cut it there. But thank you so much for your brilliant talk. There was a number of other questions we didn't get around to, so I'm really sorry for that. But please do keep sending them, and hopefully you can catch up with Nicola in the post session later on tonight.
Of course.
Well, thank you so much. Next up, we have.